Hello there, and welcome back to the Sleepy Nova Chronicles. Beneath the soil under your feet right now, something is thinking. Not in the way you think, not in the electrochemical, neuron-firing, cortex-processing way that human thought operates. In a slower way, a quieter way, a way that has no brain behind it and no nervous system to carry its signals and no skull to contain it. Because the organism doing the thinking is not contained. It spreads. Through the soil, through the leaf litter, through the decaying wood of fallen trees and the root systems of the living ones beside them, through the dark wet interior of the forest floor in a network of threads so fine and so numerous, and so deeply interwoven with every other living thing in the ecosystem that pulling one thread would, eventually, disturb them all. The fungal mycelium. The hidden kingdom that is not plant and not animal and not bacterium, that belongs to its own domain of life, the kingdom fungi, separated from every other form of life by 400 million years of independent evolutionary history and by a set of biological strategies so unlike anything that plants or animals use that the study of fungi has, over the last 3 decades of molecular biology and ecological field research, produced a picture of life on Earth that is, once properly absorbed, almost impossible to reconcile with the way most people learned to think about how the living world works. The mushroom you might see in a forest, the cap and the stem, the pale gills underneath, the specific shape and color and smell of a particular species on a particular autumn morning, is not the fungus. It is the fungus's announcement. The mushroom is the fruiting body, the reproductive structure that the fungus produces when its subterranean network of mycelium has accumulated sufficient resources, and the environmental conditions of temperature and moisture and light exposure have met the specific thresholds that the species requires to reproduce. The mushroom emerges from the soil not as the organism itself, but as the organism's broadcast signal. The structure specifically designed to release spores into the air in quantities, and from configurations that maximize the probability of at least some of those spores reaching a suitable substrate and beginning a new fungal network somewhere in the landscape. The mushroom is not the fungus growing upward. It is the fungus reaching out. And what it is reaching out from, the mycelium, the true body of the fungus, the network of microscopic threads called hyphae that permeates the soil and the wood and the living root systems of the surrounding vegetation, is something so vast, so ancient, so functionally central to the operation of every terrestrial ecosystem on Earth that once you know it is there, the forest floor you walk across will never feel the same again. You are walking on a brain. Not literally, but in the functional sense that matters for understanding what the mycelial network does. The sense in which a network of connected nodes that processes information, integrates signals from multiple sources, responds to stimuli, and coordinates the distribution of resources across a spatially extended system can be said to be performing a function analogous to what a brain performs. The mycelium does not think with neurons. It thinks with chemistry, with a specific concentration gradient-driven, signal molecule-mediated exchange of information between the nodes of a network that extends, in some cases, across hundreds of acres of forest floor and connects, through its mycorrhizal partnerships with tree roots, to every major tree in the ecosystem. This is the first thing to understand tonight, the thing that, once it settles, changes the way the soil feels under any surface you might be lying above. The majority of the Earth's land surface is underlain by living fungal networks. Not in every square centimeter. Not uniformly. But consistently enough, across enough different types of soil and enough different types of ecosystem, that the mycelium is the most abundant and the most functionally critical component of the soil ecosystem in almost every terrestrial habitat on Earth. The soil is alive. Not in the loose, metaphorical sense in which people sometimes say that. In the specific, biological, pound-for-pound measurable sense. 1 teaspoon of healthy forest soil contains approximately 1 mile of fungal hyphae. 1 mile of thread-like fungal filaments, each one between 2 and 10 micrometers in diameter, each one thinner than a human hair, packed into a volume of soil that would fit in your kitchen spoon. The density of the fungal network in the soil beneath a healthy forest is, by any physical standard you can apply to it, extraordinary. And this network is not passive. not simply occupying space in the way that a mineral or an organic residue occupies space. It is active, growing, exploring, exchanging, connecting, responding to chemical signals that tell it where resources are and where they are not, where the soil is wet and where it is dry, where a root tip has exuded the specific chemical compounds that indicate a tree is available for the mycorrhizal partnership that will sustain both organisms for the next 100 years. The mycelium is always moving. You simply cannot see it move, because it moves at the pace of biological growth, not the sudden, visible, dramatic movement of an animal, but the slow, continuous, incremental extension of a hyphal tip advancing through the soil at rates of between 1 and 40 micrometers per hour. 1 to 40 micrometers. The width of 1 to 4 human hairs per hour. So slow that no human eye watching a hyphal tip would detect movement over any period of direct observation short of time-lapse photography. But over a week, over a month, over a year, that growth accumulates into something that changes the landscape of the soil in ways that are, from the perspective of every other organism in that soil, as significant as the movement of a glacier is to the surface of a mountain. The mycelium explores, connects, transforms, in the dark below the surface, without hurry, without pause. Tonight we are going to follow the fungus, from the single spore that begins its journey, through the mechanics of how the mycelial network grows and spreads and connects to the trees it sustains, through the extraordinary chemical language of the Wood Wide Web, through the catastrophe that threatened the world's fungal networks and the recovery that is possible, to the place where all of it arrives in you, in the nutrients that the mycorrhizal network extracted from the soil and delivered to the plants that became your food, in the carbon cycle that the fungal decomposers run and that determines the composition of the air you are breathing right now, in the specific, dark, damp, unhurried, underground intelligence of the organism that has been holding the terrestrial world together for 400 million years. If this gentle journey into the hidden kingdom beneath the soil is useful to you tonight, a quiet hour of mycelium and spores and the patient, dark, networked intelligence of the most successful organism on Earth by area, then I would gently invite you to subscribe, or to leave a thought below about which part of this stays with you. It helps other people who are lying in their own dark above their own mycelial networks, also connected to the living soil without knowing it, find their way here, one spore at a time. But for now, there is only this. Allow your shoulders to soften. Let your breathing slow, not forced, not directed, just allowed to find the pace it finds when the body is still and the room is quiet and the surface beneath you is, somewhere far below, threaded through with the finest network of living filaments that the planet produces. The specific, quiet, below surface, in the dark breathing of a world that is always, gently, growing. The mycelium is already moving. Below you, in the dark. Let's begin. A single fungal spore weighs approximately 1/1000th of a gram. Hold that number for a moment before moving past it. 1/1000th of a gram. The lightest object your naked eye can detect, a fleck of dust on a sunlit windowsill, weighs approximately 1/1,000,000th of a gram. The fungal spore weighs 1,000 times less than that. Less than a bacterium. Less than a red blood cell. A particle so light that the thermal currents produced by the warmth of a single human hand can carry it upward at speed sufficient to suspend it in still air for hours. And yet this particle, this single cell, enclosed in a wall of one of the toughest biological polymers that evolution has produced, containing the complete genetic blueprint of an organism capable of spreading across hundreds of acres of forest floor and living for thousands of years, is the beginning of the most successful terrestrial organism in the history of life on Earth. The spore is released from the mushroom's gills, not gently, not passively, not simply by falling from the gill surface when gravity and a breeze combine to carry it away. Ejected. The basidiospore, the specific type of spore produced by the gills of the mushroom cap in the largest and most familiar group of fungi, the basidiomycetes, is launched from the gill surface by a mechanism of extraordinary precision that operates entirely without muscle, without any mechanical structure whatsoever, through the specific thermodynamic properties of water at a microscopic scale. On the surface of each spore-bearing cell, the basidium, a tiny drop of water forms at the base of the spore, at the junction between the spore and the cell that produced it. This drop, the Buller's drop, named for the mycologist Arthur Reginald Buller, who first described the mechanism in 1909, forms by the condensation of water vapor from the humid air of the gill chamber onto the hydrophilic surface of the spore's attachment point. As the drop grows, it reaches a critical size at which it merges with a second drop of water on the spore's flattened face. The merger releases the surface energy of the two drops, the energy that was stored in the curved surfaces of two small spheres, as a sudden, directed impulse that launches the spore away from the gill surface at an acceleration of approximately 10,000 meters per second squared. 10,000 meters per second squared. The acceleration of a bullet leaving a rifle barrel is approximately 300,000 meters per second squared. The acceleration of the spore is about 3% of a bullet's. But the spore is not moving through a gun barrel. It is moving through the still, humid air of the gill chamber of a mushroom, and for its purpose, clearing the gill surface and entering the airspace below the cap where the air currents will carry it away. An acceleration of 10,000 meters per second squared, applied to a particle weighing 1/1000th of a gram, is more than sufficient. The spore launches itself, using water as a catapult, in a chamber so dark and so humid that no human eye without a microscope would see it happen. Detectives looking at the spore ejection mechanism of basidiomycete fungi, using high-speed photography capable of capturing events at 100,000 frames per second, revealed that the entire launch, from the formation of the Buller's drop to the spore clearing the gill surface, takes approximately 100 microseconds. 1/10,000th of a second. At the moment of launch, the spore is moving at approximately 1 meter per second, which is, given that its entire trajectory through the gill chamber before it reaches the open air is approximately 2 to 4 millimeters, exactly sufficient to clear the gill without hitting the opposing gill on the other side of the chamber. 2 to 4 millimeters of trajectory, timed to 1/10,000th of a second, in the dark, inside a mushroom. The engineering precision of the spore launch is not accidental. It is the product of approximately 300 million years of evolutionary refinement of the basidiomycete fruiting body, 300 million years of trial and error, of mushrooms that launched there. Spores too weakly and hit the opposing gill and never entered the airstream, of mushrooms that launched too strongly and sent the spore into the gill above instead of the space below, of every geometric variation of gill spacing and cap orientation and Buller's drop size that a population of reproducing fungi can explore over the span of geological time. The mushroom we see today is the solution, not a perfect solution. There is no perfect solution to the engineering problem of launching a microscopic particle from a curved surface in a confined humid chamber using only the surface energy of water. But the best available solution, tested against every variant that random mutation has offered over 300 million years of continuous refinement. The spore leaves the gill, enters the airspace below the cap, and the wind takes it. A single mushroom releases between 10 million and 100 billion spores per day, depending on the species and the size of the fruiting body. 100 billion. The number of neurons in the human brain is approximately 86 billion. A large puffball mushroom, a single fruiting body, roughly the size and shape of a football, contains approximately 7 trillion spores. 7 trillion spores. In a single mushroom the size of a football. Most will never germinate. Most will land on surfaces unsuitable for fungal growth. On tarmac, on water, on rock, on soil already saturated with fungal competitors, on the leaves of plants with no mycorrhizal partnership available. The overwhelming majority will simply stop, will land, desiccate, fail to find the specific combination of temperature and moisture and substrate chemistry that germination requires, and dissolve back into the soil as organic matter, contributing nothing more to the propagation of the species than the small quantity of nutrients their decomposition provides. But some will land on suitable substrate. And one is enough. The germinating spore extends its first hypha within hours of landing on suitable substrate. The hypha, a single cell that extends itself by adding new cell wall material at its growing tip, driven by the turgor pressure of water inside the hyphal tube and guided by chemical gradients in the surrounding environment, grows toward the resources the spore's biochemistry requires. Moisture, carbon compounds, the specific mineral ions that fungal metabolism needs. The hyphal tip grows at the rates we noted in the hook, between 1 and 40 micrometers per hour, depending on the species and the conditions. At 40 micrometers per hour, the tip advances approximately 1 millimeter per day, 1 centimeter in 10 days, 1 meter in approximately 3 years. Not fast by any standard a human experience of movement applies. But the hypha does not grow alone. Within hours of the first hyphal tip extending, the growing hypha begins to branch, to produce lateral branches at regular intervals behind the tip, each branch growing in its own direction, each branch itself branching, the whole network expanding not linearly but exponentially, the area it covers doubling and redoubling as each branch produces its own branches. The mycelium grows like a thought, not in the single direction of a single idea, but in the branching, parallel, self-similar way of a mind that is considering many possibilities simultaneously, each branch a different trajectory, each branch point a decision about which direction holds the most promise, the whole network a spatial record of every exploration the organism has made since the first spore germinated. The monster of fungal biology is not the spore launch or the hyphal growth or even the scale of the mycelial network. The monster is the function, the specific, network-level, emergent function that the mycelium performs once it has grown large enough and connected to enough resources and established enough partnerships to begin doing what the mycelium, at its most fully developed, actually does. It manages the forest, not as a metaphor or a loose ecological generalization, as a specific, measurable resource allocation function that determines, across the entire forest ecosystem, which trees receive which nutrients at which times and in which quantities, and that regulates those distributions through the same kind of chemical signaling and network topology that the Internet uses to route information packets between nodes. The mycelial network is the forest's circulatory system, and the heart that drives it is the mycorrhizal symbiosis. Approximately 90% of all terrestrial plant species form mycorrhizal partnerships with fungi. 90%. The fungi that partner with plant roots are not parasites extracting nutrients from a host. They are partners in the most consequential mutualistic symbiosis in the history of terrestrial life, the specific, reciprocal, biochemically negotiated arrangement in which the fungus extends its hyphae deep into the soil, harvesting the mineral nutrients and water that the plant's roots alone cannot reach, and delivering them to the root cells in exchange for the carbon-rich sugars that the plant produces through photosynthesis, and that the fungus, living in the dark below the light zone, cannot produce for itself. The plant gets phosphorus and nitrogen and water. The fungus gets sugar. Both survive better together than either could survive alone. Your own gut microbiome operates on the same mutualistic logic, the specific, negotiated, biochemically maintained exchange between your intestinal epithelium and the several trillion microbial organisms that inhabit your digestive tract, in which the microbes digest the complex carbohydrates that your own enzymes cannot process, produce the vitamins and the short-chain fatty acids that your intestinal cells require, regulate the immune responses that protect your gut epithelium from pathogens, and receive in exchange the warm, nutrient-rich, protected environment of your intestinal tract, an environment they could not survive outside. You and your gut microbiome are running the mycorrhizal logic, in a different location, on a different scale, with different organisms, but the same fundamental principle, the mutualistic symbiosis of two organisms that provide each other with what neither can provide for itself, linked by a biochemical exchange that is so ancient and so deeply embedded in both organisms' biology that neither can function normally in its absence. The forest without its mycorrhizal fungi is a forest whose trees cannot access the phosphorus and nitrogen their growth requires, whose seedlings cannot establish themselves in the competitive, resource-limited environment of the forest floor without the fungal partnership that gives them immediate access to the mineral wealth of the soil, whose carbon and nutrient cycles run at a fraction of their full efficiency. The forest needs the fungus, as you need your gut microbiome, as the spore needs the suitable substrate, as everything, in the end, needs its partner. In the dark, below the surface, where the real work is always done. Detectives looking at the mycorrhizal networks of temperate forest ecosystems using radioactive tracer compounds injected into the phloem of individual trees revealed something that transformed the way ecologists understand the forest as a biological entity. The tracers moved, not only through the vascular system of the injected tree, which would have been expected, but through the mycelial network connecting the injected tree's roots to the roots of neighboring trees, and from those trees to their neighbors, and from those to their neighbors, propagating through the forest ecosystem in a pattern that could only be explained by the active transport of the tracer compound through the fungal network. The carbon that the large trees fixed through photosynthesis was moving through the mycelial network to the smaller trees in the understory, to the seedlings that were growing in the deep shade where photosynthesis was insufficient to sustain their growth. The forest was feeding its young through the fungal network. The specific, measured, tracer-verified flow of carbon from the canopy trees to the shade-bound seedlings via the mycorrhizal mycelium is the most direct experimental evidence available for the hypothesis, now accepted by most forest ecologists, that the mycelial network functions as a resource-sharing system that maintains the forest community's overall productivity by redistributing surplus resources from the large and well-lit to the small and shade-limited. The forest is not a collection of competing individuals. It is a community with a shared circulatory system. And the circulatory system is the mycelium. Your own social network runs on the same principle. The specific, informal, mostly invisible flow of resources and support and information between the connected members of a community, from those with surplus toward those with need, sustained not by any formal redistributive mechanism but by the simple fact of connection, the fact that when you are connected to another person, the resources you each have access to become, in some meaningful sense, resources that both of you can draw on. The mycelium does not decide to share resources. It simply provides the connection through which sharing becomes the path of least resistance. The forest provides the sharing. The mycelium provides the path. When a tree is wounded, it sends a message. Not a message in the linguistic sense, not a signal encoded in arbitrary symbols that require a shared interpretive framework to decode. A message in the chemical sense, a specific compound released from the site of the wound into the vascular system of the tree, and from there into the mycorrhizal network, connecting the tree's roots to the mycelium of its fungal partner, and through that partner to the roots of every other tree connected to the same mycelial network. The message travels through the soil, and the trees that receive it respond. Not slowly, not after a season of biological adjustment. Within hours of receiving the chemical signal from a wounded neighbor, undamaged trees in the connected network upregulate their production of the specific defensive compounds, the terpenes, the tannins, the alkaloids, that make their leaves and bark less palatable to whatever herbivore or pathogen is attacking the source tree. They do not wait to be attacked. They prepare in advance. On the basis of a warning received through a network they cannot see, from a neighbor they have no direct sensory contact with, in a language that has no words but carries, in its specific chemistry, the precise information that their survival requires. This is the Wood Wide Web, not a metaphor borrowed from human technology and applied loosely to a biological phenomenon that happens to involve a network structure. A specific, empirically documented, chemically characterized system of below-ground communication that connects the trees of a forest ecosystem through the mycelial network of their shared fungal partners, and that allows the transmission of ecologically significant information, warnings of herbivore attack, signals of carbon deficit, communications of resource availability, across distances and between individuals that would, in the absence of the mycelial network, have no means of direct communication at all. Detectives looking at the specific chemical compounds involved in mycorrhizal network communication using Gauss chromatography and mass spectrometry identified a vocabulary. Not a human vocabulary, not a vocabulary of symbols that can be translated into meaning without loss, but a functional vocabulary of compounds whose presence in the network soil solution correlates with specific ecological states and produces specific responses in the trees that detect them. The signal compound for herbivore attack is different from the signal compound for fungal pathogen attack. The signal compound for carbon deficiency in a neighboring tree is different from the signal compound for phosphorus deficiency. Each message has its own molecule. Each molecule has its own response. The vocabulary is small by the standards of human language, perhaps dozens of distinguishable signal compounds rather than the hundreds of thousands of words that a human language provides. But the information it carries is not small. The information it carries is the specific, timely, ecologically actionable intelligence that allows a forest community to respond to threats collectively rather than individually, to mobilize its defenses before the attack reaches any given individual, to redistribute resources before any given individual is depleted, to function as a distributed, networked, chemically communicating organism rather than a collection of isolated competitors. The forest is not competing. It is coordinating. And the coordinator is the mycelium. Your own immune system coordinates by the same chemical language logic. When a cell in your body encounters a pathogen, when a macrophage engulfs a bacterium and presents its antigens to the adaptive immune system, it releases specific cytokines, small signaling proteins whose presence in the local tissue chemistry carries specific information about the nature of the threat and the specific immune response that the threat requires. Interleukin-1 signals that bacteria are present and macrophage activation is underway. Interleukin-4 signals that parasite defense is needed. Interferon gamma signals that viral infection has been detected and cytotoxic T-cell activation should proceed. Each cytokine is a word in the immune system's chemical vocabulary. Each word activates a specific response. The immune system communicates by molecular message, as the forest communicates by molecular message. Both systems maintaining the health of a complex organism, you in one case, the forest in the other, through the specific, chemistry-mediated, network-distributed coordination of defensive responses that no single cell and no single tree could mount alone. Now follow the mycelium to its most extraordinary function, not the mycorrhizal partnership, not the resource sharing, not the chemical communication, but the specific work that the saprotrophic fungi perform. The decomposers, the organisms that take the dead material of the forest, the fallen trees, the leaf litter, the animal corpses, the everything that living things produce and leave behind when they die, and convert it back into the mineral nutrients that the next generation of living things can use. The saprotrophic fungi are the forest's recycling system, and the specific enzymatic tools they use to perform that recycling are, in their chemical sophistication and their substrate range, unlike anything that any other organism on Earth produces. Lignin is the most abundant organic polymer on Earth after cellulose. It is also the most chemically resistant. The specific three-dimensional, aromatic, cross-linked polymer structure of lignin, evolved by plants approximately 350 million years ago as the structural material that makes wood hard and wood-like rather than soft and grass-like, is so resistant to the enzymatic attack of most organisms that it was essentially indestructible for approximately 60 million years after its first appearance in the fossil record. Not destroyed by bacteria, not degraded by most fungi, simply accumulating in the soils of the Carboniferous period, in the thick, anoxic peat deposits that the falling forests of the coal swamps were building over 60 million years of lignin accumulation that nothing in the ecosystem had yet evolved the tools to break down. Those deposits are the coal seams. The coal that the Industrial Revolution burned was primarily the accumulated lignin of 60 million years of undecomposed Carboniferous forest. 60 million years of the world's most abundant structural polymer, accumulating because nothing could eat it, until the white rot fungi evolved. The white rot fungi, a diverse group of basidiomycetes, the same broad group that includes the familiar mushrooms of the forest floor, evolved, approximately 300 million years ago, the specific oxidative enzymes, the lactases and the lignin peroxidases and the manganese peroxidases, that can attack the aromatic rings of the lignin polymer, break its cross-links, and convert the most chemically resistant natural material in the terrestrial biosphere into the small organic molecules. That bacteria can then metabolize completely. The white rot fungi solved the lignin problem, and in solving it, they transformed the carbon cycle of the entire planet, ending the 60 million year accumulation of undecomposed lignin, restoring the carbon cycling of forest ecosystems to something close to its modern efficiency, and creating, through the opening of new ecological niches in the decomposer food web, the conditions for the extraordinary diversification of forest ecosystems that characterizes the last 300 million years of terrestrial life. The white rot fungi saved the world from being buried in wood. The monster of fungal decomposition is this function, the specific enzymatic capacity to dissolve the most recalcitrant organic material in the terrestrial biosphere and return its constituent elements to the soil in forms that other organisms can use. Not the dramatic monster of sudden destruction. The white rot fungus does not destroy wood in any time frame that a human observer would call fast. It dissolves it, over years and decades, through the patient application of enzymes that few other organisms on Earth possess to a substrate that, without those enzymes, would simply accumulate indefinitely. The patience of the dissolving is what makes it powerful. A fallen oak tree that begins its decomposition in a temperate forest can take between 50 and 100 years to be fully processed by the fungal community that colonizes it. 50 to 100 years of progressive enzymatic dissolution, the fungal hyphae advancing through the wood cell by cell, breaking down the cellulose and the lignin, releasing the nutrients locked in the wood structure, creating in the process the specific microhabitat of the decaying log, the soft, damp, dark interior with its specific temperature and moisture and chemical character that supports its own community of organisms, the beetles and the millipedes and the bacteria and the mosses and the seedlings that germinate in the rich substrate of the advanced decay stages. The fallen tree becomes, over a century of fungal digestion, the richest habitat in the forest floor. Death, processed by patience, becomes abundance. Your body runs the same transformation during sleep. The metabolic waste products of the day, the misfolded proteins, the damaged lipids, the accumulated cellular debris that the day's biochemical activity produces, are processed during sleep by the glymphatic system, the brain's waste clearance mechanism that activates specifically during slow-wave sleep and flushes the interstitial fluid through the spaces between the neurons, carrying the day's metabolic waste toward the blood-brain barrier and the lymphatic system and ultimately out of the brain entirely. The dead material of the day's cognitive work is broken down during the night by the brain's equivalent of the fungal decomposer. The glymphatic system does for the brain what the white rot fungi do for the fallen oak. It dissolves the accumulated residue of the day's activity and converts it from the inert, potentially harmful form of metabolic waste into the cleared, clean, functionally available substrate that tomorrow's cognitive work requires. Sleep is the brain's decomposition, not of the brain itself, not the destruction of the architecture that the day built. The decomposition of the day's waste. The patient, enzymatic, biochemically specific dissolution of the accumulated debris that living produces, conducted in the dark, in the quiet, while the organism that produced the debris is temporarily unavailable for other business. The forest floor is doing this right now. Below the surface. In the damp dark of the soil. The mycelium advancing through the fallen wood, the enzymes dissolving the lignin, the nutrients returning to the soil water, the carbon cycling back through the roots of the living trees into the atmosphere and back again. The world is decomposing itself tonight. as your brain is decomposing its day. Both of them patient. Both of them processing in the dark. Both of them converting the residue of what was into the substrate of what will be. Quietly. Without hurry. Without waste. 100 years ago, a cargo ship arrived in New York Harbor carrying timber from Asia. The timber was infected. Not visibly. Not in any way that the customs inspectors of the early 20th century, working without molecular diagnostics, without the polymerase chain reaction, without the rapid genetic identification tools that modern biosecurity uses to detect plant pathogens at ports of entry, could have identified from the appearance of the wood. The infection was microscopic, dormant, latent in the bark of the timber in the form of the spores of a fungus called Cryphonectria parasitica, the chestnut blight fungus, that had co-evolved over millions of years with the Asian chestnut trees of its native range and that had, in that co-evolution, reached the specific accommodation between pathogen and host that defines a stable parasitic relationship. The fungus infecting the tree without killing it, the tree tolerating the fungus without eliminating it. The American chestnut had never met this fungus. It had no evolved defense against it. The spores entered the forest. And within 50 years, the American chestnut, the dominant canopy tree of the eastern North American forest, constituting approximately 25% of the total canopy cover of the forests from Maine to Georgia, producing the most important mast crop in the eastern forest ecosystem, providing the food source that sustained the passenger pigeon and the white-tailed deer and the black bear and dozens of other species of the Appalachian forest community, was functionally extinct as a forest tree. Functionally extinct. Not gone from the landscape. The American chestnut's root system survived the blight. The fungus attacked only the bark above the soil surface, the cambium above the graft union, the living tissue of the trunk and the branches, and could not reach the root system below the soil line. The roots persisted. They continue to send up new shoots from the root crown to this day, shoots that grow to sapling height, that reach the diameter at which the blight spores can infect them, and that then die back to the root crown as the infection girdles the bark and kills the cambium above the infection site. The American chestnut sprouts, grows to a sapling, encounters the blight, dies back, sprouts again, has been doing this in the forests of the eastern United States for 100 years. Not dead, not alive in the full sense that a 50-meter, 2-ton canopy tree constitutes alive, trapped in a cycle of growth and death that the persistent root system sustains and that the persistent blight prevents from completing. The monster of the chestnut blight is not the fungus itself, not Cryphonectria parasitica as an organism, which is, in its own ecological context, simply a fungus doing what fungi do. The monster is the function of the introduction, the specific, irreversible, ecosystem-level consequence of moving a pathogen from the host community that had co-evolved the capacity to tolerate it into a host community that had not. The naive host. The American chestnut had no defense against the blight because it had never needed one. Its evolutionary history, the millions of years of selection pressure that shaped the specific immune responses of its bark tissue, contained no encounter with Cryphonectria parasitica, no period of co-evolutionary negotiation between the tree's defenses and the fungus's virulence, no accumulated population of survivors who had demonstrated some capacity to tolerate the infection and passed that capacity to their offspring. It was perfectly defenseless. As the American chestnut was defenseless, so were the mycorrhizal networks that had been built around it over centuries of forest development. The specific fungal community that had co-evolved with the American chestnut, the specific mycorrhizal fungi that had formed partnerships with its roots over centuries, that had calibrated their resource exchange chemistry to the specific metabolic needs of the chestnut, that had organized the nutrient flows of the eastern forest around the chestnut's dominance of the canopy, lost its primary partner. When the chestnut died, the network changed, not collapsed. The mycorrhizal networks of the eastern forest were diverse enough that the loss of one major host tree, even the dominant canopy species, did not eliminate the network. But it changed its character. The specific flows of carbon and nutrients that the chestnut had been driving through the network were reduced. The ecological space that the chestnut had occupied was available for colonization by other species, primarily the oaks and the hickories that had been growing in the chestnut's shade and that expanded to fill the canopy gaps the chestnut's death created. And the mycorrhizal networks reorganized, partially, incompletely, over decades of ecological succession, around the new community of dominant trees. The forest survived, but not as what it had been. Detectives looking at the ecological history of the eastern North American forest using pollen records from lake sediments and wood fragments from archaeological sites and the written accounts of early European settlers revealed the specific character of what was lost. Not just the tree itself, but the specific ecological services the tree had provided and that no successor species has fully replaced. The chestnut produced its mass crop, its crop of large, starchy, nutritious nuts, in years when the oaks produced nothing, providing the interannual dietary redundancy that buffered the animal communities of the forest against the mast failure years that oak-dominated forests are prone to. The chestnut was the forest's food security system, and it is gone. Now multiply the chestnut blight across the full catalogue of fungal diseases currently threatening the world's forest and agricultural ecosystems. The Dutch elm disease that has killed the majority of the elm trees in Europe and North America. The ash dieback that is eliminating the ash trees of European forests at rates that elm disease never achieved. The sudden oak death that is devastating oak populations on the Pacific coast of North America. The pine pitch canker that threatens the pine forests of 5 continents. The Panama disease, Fusarium wilt, that twice in the last century has brought the global banana industry to the edge of extinction by infecting the specific cultivars on which commercial production depends. All of them fungal. All of them introduced pathogens encountering naive hosts. All of them producing, in the ecosystems they enter, the same cascading consequence. The loss of a dominant or keystone species, the reorganization of the mycorrhizal network around its absence, the reduction of the ecological complexity and the resource exchange efficiency that the lost species and its fungal partners had built over centuries of coevolution. The catastrophe of fungal disease is the catastrophe of the broken network. Not the loss of the individual tree. The individual tree is replaceable by the seeds and seedlings of its own species, or by the succession of other species into the vacated ecological space. The loss of the network architecture that the individual tree had built and maintained, the specific pattern of mycorrhizal connections, the specific flows of resources and signals that ran through those connections, the specific ecological function that the network's topology performed, is not so easily replaced. Networks have memory. The memory of a mycorrhizal network is the specific pattern of its connections. Which trees are connected to which fungal species are connected to which soil patches, which pathways carry carbon and which carry nitrogen and which carry the chemical warning signals of herbivore attack. This memory is not stored in any individual node. It is distributed across the entire network, expressed in the pattern of connections rather than in any individual connection. When a node is lost, when a tree dies and its mycorrhizal connections are severed, the network does not simply lose one connection. It loses the specific contribution that that node made to every network function that ran through it. The resource flows that pass through that node must find alternative routes or cease. The warning signals that would have been relayed through that node must propagate by different paths or fail to propagate at all. The network is restructured around the absence, not always with the same efficiency, not always with the same ecological function. But it is restructured. And here, in the restructuring, in the network's persistent capacity to reorganize around loss and find new pathways for the functions the lost nodes performed, is the wonder without anxiety reframe that this movement requires. The mycelial network does not mourn its lost nodes. It grows new connections. The forest that lost its chestnuts grew new canopy trees and new mycorrhizal partnerships. The forest that lost its elms grew new understory light conditions and new ground-level communities. The forest that is currently losing its ashes is already showing, In the ecologically progressive areas where the disease has been present longest, the first signs of the community reorganization that will eventually produce a new forest composition, different from the ash-dominated forest that preceded it, structured around whatever species survive and thrive in the new light and soil conditions that the ash's absence creates. The catastrophe clears the space. The network fills the space with whatever is available, with whatever the spores in the soil and the seeds in the seed bank and the surviving mycorrhizal fungi can produce in the conditions the catastrophe leaves behind. The forest has been doing this for 400 million years, through mass extinctions and glaciations and volcanic winters, and the full catalogue of catastrophes that the geological record preserves. It has always found a way to fill the space, because the mycelium is still there, in the soil, growing, connecting, finding the path of least resistance between the resources that are available and the roots that need them, not grieving the lost network, building the new one. Your own neural networks perform the same reorganization around loss. When a connection is broken, when a relationship ends, when a skill atrophies from disuse, when a pathway that the brain relied on for a specific cognitive function is disrupted, the network does not simply carry the gap. It reroutes. Other pathways strengthen to carry the function that the lost pathway performed. New connections form in the regions that neighbor the disrupted circuit. The specific, experience-dependent plasticity of the brain, the capacity to modify the strength and the pattern of its connections in response to change demands, is the neural equivalent of the mycelium growing new hyphae around the obstacle that the environment presents. The brain grows around its losses, as the forest grows around its. Both of them patient, both of them rebuilding in the dark, both of them converting the catastrophe into the substrate of the next growth. One connection at a time, in the soil, in the dark, in the slow, below-conscious, unhurried way that the real work of recovery always proceeds. There is fungal material in your body right now. Not an infection, not a pathogen. The specific, chemically active, biologically essential products of fungal metabolism that have entered your body through your food and your gut microbiome and the air you breathe. The ergosterol that the sun converts to vitamin D2 in your skin when you eat fungal food exposed to UV light. The beta-glucans that stimulate the innate immune responses of your intestinal epithelium. The specific B vitamins that the fungi in your gut microbiome produce and that your intestinal cells absorb. You are running on fungal chemistry. The vitamin D in your blood, the specific steroid hormone that regulates calcium absorption, that maintains bone density, that modulates the immune system, that has been associated in study after study with the regulation of mood and the reduction of inflammatory disease risk, is produced in your skin from a precursor compound called ergosterol. Ergosterol is a fungal compound. Your skin cells do not produce ergosterol. They convert it from the dietary ergosterol that you absorb from the fungi in your food, or from the small amounts of ergosterol in the cell membranes of other organisms in your diet, into the pre-vitamin D3 that UV light then converts into the active vitamin. The sun activates the fungus that the food provided, and the result runs your bones. Detectives looking at the relationship between dietary fungal consumption and human vitamin D status in populations with limited sun exposure, populations in northern latitudes, populations who work indoors, populations whose vitamin D production from sun exposure is insufficient for their physiological needs, revealed that the specific form of vitamin D provided by dietary fungi, vitamin D2, ergocalciferol, is biologically active in humans and contributes meaningfully to vitamin D status. In populations where sun exposure alone is insufficient, the fungus has been supplementing human biology for as long as humans have been foraging in forests. Long before anyone knew what vitamin D was. Long before anyone understood why the people who ate mushrooms seemed, in certain seasons and certain latitudes, to fare better than the people who did not. The fungus knew what the body needed, not consciously, not with any awareness of human nutritional requirements. Through the evolutionary accident of producing a compound that happened to be useful to every organism that consumed it, that happened to fit into the biochemical pathways of a wide range of organisms because those pathways had evolved in an environment where the fungus was everywhere and its compounds were always present in the food. You are running, in part, on the chemistry of an organism that has been preparing your food for 400 million years. Now follow the fungal network all the way to the place where it arrives most directly in you, not through the chemistry of what you eat, but through the specific biological architecture of the organization you are part of. You are a mycelium, not metaphorically assembled from a loose network analogy. Structurally, functionally, architecturally. Your nervous system, the 100 billion neurons connected by approximately 100 trillion synapses into the specific, branching, parallel processing, signal-propagating network that constitutes your brain and spinal cord and peripheral nervous system, is a mycelial network expressed in biological tissue. The branching pattern of a hyphal network and the branching pattern of a neural dendritic tree are not merely visually similar. They are solutions to the same mathematical problem, the problem of maximizing the surface area available for exchange while minimizing the material cost of the structure that provides that surface. The fractal branching that the hypha uses to explore the soil and the fractal branching that the dendrite uses to receive synaptic inputs from its neighbors are both solutions to the same optimization problem, arrived at independently by organisms separated by 400 million years of evolutionary history. Evolution found the same shape, because the same shape solves the same problem. The mycelium and your dendrites are convergent solutions to the problem of connecting as much as possible with as little material as possible. The fundamental engineering constraint of any biological network that must cover large areas and process large volumes of information without the weight and the metabolic cost of solid tissue. Your brain is efficient. The mycelium is efficient. Both for the same reason. The branching works. And right now, in the dark behind your eyes, in the warm enclosure of your skull, in the specific, increasingly slow and synchronized electrical rhythms of a brain that is approaching the threshold of sleep, your mycelium is doing what the forest's mycelium does in the hours after the day's photosynthesis has ended, and the carbon supply has been reduced, and the network is running on the reserves it accumulated during the light. It is maintaining, not growing new connections. Not in the dramatic, experience-dependent way that learning grows new synaptic connections during the day. Maintaining the specific, cellular, molecular maintenance of the synaptic structures that the day built, the turnover of the synaptic proteins that keep the connection functional, the recycling of the neurotransmitter vesicles that the day's firing depleted, the restoration of the ion gradients that the action potentials of waking consumed. Maintenance. In the dark, while the network is quiet enough that the maintenance can proceed without competing with the demands of active transmission. The forest's mycelium performs the same maintenance in the dark, the specific, cellular, enzymatic maintenance of the hyphal network that keeps it functional between the resource pulses of the growing season. The repair of hyphal walls damaged by soil chemistry. The recycling of the hyphal contents of old, non-productive parts of the network into the growing tips of new, exploratory hyphae. The continuous, low-level, metabolically expensive maintenance of a network that is, in its full extent, the largest and most complex biological structure in the ecosystem. Both networks maintaining themselves in the dark. Both running on reserves accumulated during the day. Both preparing for the next day's growth. Now follow the spore, not to its germination, not to the mycelium it will eventually become, but to the journey it makes between the cap of the mushroom and the soil of the forest floor. The journey that takes it from the specific point where it was ejected by the buller's drop mechanism into the still air of the gill chamber, through the turbulent, unpredictable, wind-driven airspace above the forest floor, to wherever it eventually lands. The spore does not know where it is going. It does not navigate. It does not steer. It does not have a destination it is trying to reach, or a route it is following, or a preference among the infinite possible landing sites that the wind will choose for it. It is simply present in the airstream, carried by whatever current exists at the moment it enters the air, deposited wherever that current dissipates or wherever an obstacle presents itself. And yet the dispersion strategy works. Not because every spore finds a suitable substrate. We established in Movement 2 that the overwhelming majority do not. But because the production of billions of spores from a single mushroom, released over the course of days and weeks, distributed by the wind into every direction and every distance that the wind can reach, ensures that the small fraction of spores that happen to land on suitable substrate is, in absolute number, sufficient to establish new fungal networks wherever the conditions allow. The strategy is not precision. It is abundance. Not directing every effort toward the single most promising target. Distributing effort across the entire landscape, trusting that the abundance of attempts will produce, in the fraction that succeed, enough new growth to sustain the species. Your own life runs on the same strategy in the specific domain of creative and intellectual effort. Not every idea you have is worth pursuing. Not every project you begin will produce something significant. Not every conversation you initiate will lead somewhere important. The abundance strategy, generating many attempts, trusting that the small fraction that succeed will justify the effort of all the ones that did not, is the strategy that the most productive creative lives use, knowingly or not. Not because they are careless about quality. Because they understand, as the fungus understands without understanding anything at all, that the filtering for quality happens after the generation of abundance, that you cannot know in advance which spore will land on the right substrate, and the attempt to predict which ones will succeed and produce only those ones is, in the end, less productive than producing all of them and letting the substrate decide. The mycelium does not audition its spores. It releases them, and trusts the world. You have been releasing something today, some version of effort or attention or care or creativity that went out into the world and landed wherever the currents of the day took it. Some of it will have landed on suitable substrate. Some will have found the specific combination of conditions that allows germination and growth and the beginning of something new. Most will not. Most will have landed on tarmac or stone or on the wrong soil or at the wrong time. This is not failure. This is the spore strategy. This is how the forest spreads. 1 billion spores per day. Most of them lost. Some of them everything. If this journey through the hidden kingdom of the fungal world, through the ejected spore and the growing hypha and the wood wide web and the chestnut blight and the white rot fungi dissolving their way through 60 million years of accumulated lignin, has been useful to you tonight, has helped the day's effort settle into the patient, dark, below-surface rest that the network requires between its days of growth, then I would gently invite you to subscribe, or to leave a thought below about which part of this stayed with you longest. Whether it was the 1 mile of hyphae in a teaspoon of soil, or the buller's drop launching the spore in 1/10,000th of a second, or the 7 trillion spores in a football-sized puffball, or the message that the wounded tree sends through the mycelium to warn its neighbors, or the white rot fungi dissolving the coal that 60 million years of lignin made, or simply the knowledge that the vitamin D in your blood right now began as a fungal compound in the food that the forest grew. It helps other people find their way here, other nodes in their own mycelial networks, also maintaining in the dark, also preparing for tomorrow's growth. One hypha at a time. And if sleep is still arriving slowly, if the network is still transmitting at a rate the night does not require, there is another journey waiting. Another hidden world below the surface. Another piece of deep biology that has been running the planet since before animals existed, told at this same unhurried pace, in this same quiet voice, carrying you somewhere darker and softer and older than anything that needed attending to today. It will be there when you need it. But for now, feel the weight of your body where it meets the surface beneath you. Below that surface, below the floor, below the foundation, below the soil that the building sits on, the mycelium is there, threading through the dark, damp earth. One hypha at a time, not rushing, not competing with the night's quiet, just growing in the direction of what it needs, in the way that living things grow, toward the resources. away from the obstacles. In the dark, allow your shoulders to soften now, the way the hyphal tip softens when it enters a region of high moisture, no longer pressing against resistance, finding instead the yielding substrate that its chemistry has been seeking, the specific release of a growing tip that has finally found the right conditions, the softening that happens when the environment provides what the organism needs. Allow your jaw to unclench. Allow your hands to open. Allow the thoughts that are still branching at the edge of your awareness, still extending new hyphae into the day's unanswered questions, still exploring the soil of tomorrow's concerns, to find the boundary of the night, to reach the substrate that does not support growth, to stop extending into it, to draw back to the network, to maintain. Tonight is a maintenance night, not a growth night, not a branching night, a night for the network to run on its reserves, to repair what the day used, to restore what the day depleted, to sit in the dark soil of the night and do the patient, below conscious, metabolically expensive work of keeping the connections alive. The mycelium is doing this right now. Below you, in the dark, 1 mile of hyphae in a teaspoon of soil, all of it maintaining, all of it patient, all of it waiting for the conditions that will allow the next day's growth. Beneath the soil right now, something is thinking, not in the way you think, in the slower way, the quieter way, the way that has no brain and no skull and no boundary, because it spreads through the dark, through the damp, through the living root and the fallen wood and the patient, below-surface geography of a world that has been here 400 million years longer than anything that worried you today. You are part of that network tonight, connected, maintained, in the dark, growing, tomorrow, resting now. Sleep well, and good night.